Okay, quickly going to the first slide. I just want to make sure that everybody understands. At the heart of genetic engineering is the ability to isolate and manipulate DNA. Isolate and manipulate DNA. And so there are four major steps that are involved in isolating and manipulating. And the first step is identification of the molecule. When we talk about the molecule, it is usually the protein molecule, right? And an example of that is insulin. Insulin is a protein that our body normally produces. And so our body, human bodies, have the gene for producing insulin. Now remember, gene is the DNA, and it's not protein. It's the instructions for making the protein, right? So our body has the gene, which is a piece of DNA, that has the instructions to make the insulin. So what we have to do is to first isolate those instructions from the human cells, okay? So we need to be able to cut out the gene from the human genome. And next, so once you isolate the instructions, you need to be able to manipulate the DNA instructions. So essentially, you have to take this gene for insulin. Now I'm just giving insulin as an example, but it can be any protein, right? And you need to be able to transfer that into another cell. It could be E. coli. So another cell could be E. coli, right? So that the gene needs to be able, we need to be able to place the gene into the E. coli and be able to grow the E. coli so that they can E. coli can then make protein, the insulin protein. Okay, so that's what we mean by manipulation of the DNA instructions. We need to be able to cut them from one source and paste them in another. Okay, and once you have done that, you can literally grow this E. coli cell in culture in bioreactors, and they will be producing a lot of the product, which is insulin. And so we need then to be able to harvest the molecule or the product, test it, and market it. So this is the chronological order for genetic engineering. Okay, so how do we manipulate the genes? Well, it turns out that we have discovered some really useful tools. Okay, so these are some of the tools for genetic engineering. Just like, you know, what if you wanted to cut a piece of cloth? If you wanted to cut a piece of cloth, you would need scissors, right? Same way, when you need to excuse me, when you need to cut DNA, we have these molecular scissors. So they are tools, just like scissor is a tool. We have molecular scissors that we use to cut the DNA. So, and these things are called restriction endonucleases. Restriction endonucleases. Or for short, you can also call them restriction enzymes. Now, the fact that they are enzymes should tell you that they are proteins, right? Enzymes are usually proteins. So, restriction endonucleases or restriction enzymes, they are proteins, and people have isolated them from bacteria. Now, why do bacteria have these enzymes in them? And what these enzymes do is they actually cut the DNA in specific places, right? These restriction enzymes cut DNA in very, very specific places. They have what we call restriction sites. So what happens with these enzymes is they recognize a certain sequence of DNA. Okay, and they will only cut the DNA at that particular site. Okay, and these bacteria have come up with these restriction enzymes as a defense mechanism against viruses. So remember, viruses have their own DNA, right? Viruses have their own DNA. So if a virus infects a bacterium, then the bacterium will use this restriction enzyme and cut up the virus so that it doesn't, it's a defense against the virus. Now, you might ask the question, well, the bacterium has its own DNA, right? So how come the restriction enzyme doesn't cut up the DNA of the bacteria? Well, what the bacteria do is they attach what are called methyl groups, right? So they methylate their own DNA. That means, so within the bacteria, the DNA that bacteria has, so let's say that DNA is this, ATGC combination. It's a sequence, right? So here is the DNA sequence. I'm just putting up a hypothetical one over here. Okay, so this is my double-stranded DNA. So what the bacteria do is they actually attach methyl groups. Methyl group is a CH3 group. Two parts of the DNA. So this is what we mean by methylation, right? And by attaching these methyl groups to the DNA, these methyl groups protect the DNA from restriction enzymes. Okay, so the bacteria that are producing restriction enzyme, they methylate their own DNA, so they protect themselves, their own DNA, from their own restriction enzymes. But if the viruses come in, then they can chop up the DNA of the virus. Does that make sense? So this is how some bacteria have evolved defense mechanisms against viruses. Okay, so now we have discovered lots of these restriction enzymes. We have discovered lots of the restriction enzymes. So more than 200 commercially available restriction enzymes. There are more than 200 commercially available restriction enzymes, and we have found more than 100 different recognition sequences. Okay, so you can imagine that you literally have a toolbox. You have a toolbox of these molecular scissors that are very precise. They cut in only specific places. They don't cut randomly, but only in specific places. Now you have the ability to go in and cut the DNA that you want in very, very specific places. Okay, so really neat thing, the restriction enzymes, restriction endonucleases. Okay, so any questions about restriction enzymes? Remember, they're enzymes. That means they're proteins, and they interact with DNA to cut them in specific places. Okay. Okay. So what's so nice about these enzymes? Well, there are three different types: types 1, 2, and 3. Okay. Excuse me. Types 1 and 3 are not commercially useful because their restriction sites are away from the recognition sequence. Okay, so remember that every restriction enzyme has a sequence of DNA that it recognizes. Well, it recognizes one part of the sequence, one part of the DNA, but it cuts in. So here is the DNA. So it will recognize one part. It will attach itself over here, but it will cut over here. So that's not nice. You want your recognition sequence to be at the same place where the restriction site is, right? So restriction site is away from recognition sequence for these two types of restriction enzymes. They also need ATP for cleavage. To cut the DNA, these types 1 and 3, need energy. That means you have to provide ATP. And that's not nice either. And finally, they have some DNA modification activity. That means they actually change the sequence of the DNA while they are cutting it, which is not nice either. So that's why we don't use types 1 and 3 restriction enzymes. So most of the commercial enzymes are type 2. Type 2. So what's nice about type 2? Their restriction sites are within or close to the recognition sequence. Do we know the difference between a restriction site and a recognition sequence? Can you tell me if you can tell the difference between a restriction site? So the restriction site is the sequence that is actually cut. Okay, that is actually cut. And the recognition sequence is the sequence of DNA which the enzyme binds to. So as an analogy, I can give you an analogy of, again, cutting a cloth, right? You have a piece of cloth and you want to cut it over here, but you hold it over here and over here, right? You hold it with your hand in two places, and then you are going to cut it over here. Okay, so where you hold it is your recognition site, and where you cut it is the restriction site. Does that make sense? Okay, I'm assuming it makes sense. Okay. All right, so that's one advantage. The restriction site for type 2 enzymes is within or close to the recognition sequence. And they don't need ATP. They just need some divalent ion. In this case, I'm saying magnesium as a cofactor. So as long as you provide the cofactor, they do their restriction activity. They do their cutting easily. The cuts are predictable and consistent. You can tell where the cuts are going to be, not random, right? Not random. And finally, there is no modification of the target DNA. So the target DNA stays the same. So that's important. You want your gene to be intact, right? So that's why most of the commercial enzymes are type 2. Okay? Okay. So you will, when we talk about restriction enzymes, you will hear names like Hindi3, okay, or EcoR1, for example. Here is one commonly used restriction enzyme, EcoR1. So there is a certain naming convention that is used for naming the restriction enzyme. So the first letter refers to the genus. Okay, so in this case it is Escherichia. Okay, the next two letters refer to the species. In this case, it's coli, right? Escherichia coli, E. coli, right? And then the fourth letter refers to the strain of that bacterium, the strain of the bacterium. So in this case, it is RY13. So that's why R, okay? Echo, R. And then finally, the last number, the Roman numeral, it refers to the sequence when it was actually identified. So if I say Echo R2 would be the second restriction enzyme identified from E. coli strain RY13. Does that make sense? Okay, so this actually refers to the to when the chronological sequence when it was identified, the way it was identified. Okay, so for example, Hindi 3, this refers to the Haemophilus influenzae, Haemophilus influenzae. D refers to the strain. I don't remember the strain. And then 3 is the third enzyme that was isolated from Haemophilus influenzae. Okay, are we clear about the naming convention for restriction enzyme? So restriction enzymes are named after the bacteria that they were isolated from, their genus, the species, the specific strain, and then the order in which it was isolated. Okay. Any question about the naming convention for restriction enzymes? Okay. So Victoria is asking, what about SAL1? I'm not familiar with SAL1, but I am assuming S has to do with the salmonella. There you go. And then 1 is the first one that was identified. Okay. All right. So unless there is a specific strain, they may not put in the strain identification. Okay. All right. So clear the page and moving forward. Okay. So now let's talk about the recognition sequences and cuts. So recognition sequences for these restriction enzymes are often palindromic. Palindromic means they are the same this way and that way, right? So you can see that here is a recognition sequence. It goes GGATCC this way and GGATCC this way. So it's palindromic. And oftentimes, when the restriction enzymes cut the recognition sequence, it leads to blunt or sticky ends. In this case, this is a sticky end. Okay, sticky. What do we mean by sticky end? You see that over here, this is a hanging end. This is an overhang right here, right? What's going to happen? If you have these ends like this, it becomes easy for a nucleotide to come in and bind. So it becomes easy for this to bind to another G, this to bind to another A, this to bind to another T, and this to bind to another C. So this is why we call these sticky ends. These are sticky ends, right? And the neat thing is, let's say that you cut a piece of DNA with one particular restriction enzyme. It's always going to create the same sticky ends. This enzyme is going to create the CTAG and the GATC ends, right? So imagine that here is my DNA that has been cut, and this one has the CTAG end, and this one has the GATC end. You get that? Okay. Now, you use the same restriction enzyme to cut something else, another piece of DNA. You use the same restriction enzyme to cut another piece of DNA, and guess what? That is also going to create the same sticky ends, right? Okay. Now imagine that this DNA is from human source. Okay, and this DNA is an E. coli plasmid. Plasmid is a piece of DNA from E. coli, right? So guess what? We have created the same sticky ends both from the human DNA and the E. coli DNA. Does that make sense? Now, if you mix these together, this DNA can go and literally bind to this DNA because they have corresponding sticky ends, right? Because this G is going to bind to the C, A is going to bind to this T, T to the A, and C to the G. Does that make sense? So you have literally, by using the same molecular scissors, you have cut two different pieces of DNA but created ends that will match perfectly. It's almost like Velcro, right? You have created two ends with Velcro that matches. And so you are now able to take DNA from one source and literally are able to paste it into another source, and it matches perfectly. Okay, so that's the beauty of the restriction enzymes. They cut it in a way that makes nice sticky ends that are perfectly matched, that can be perfectly matched. Okay, let me know if you are understanding it or you have questions about this sticky end business, sticky business. Okay, now sometimes you create the restriction enzymes would create sticky ends. Other times they would create blunt ends as well, and that is also useful in some cases, creating blunt ends. Okay, so in that case, so you might have a sequence like A, G, T, C, C, and you have C, C. Let's call it A. Oops. Scratch that. Anyway, it would literally, so this would be T, C, A, G, G, T, and it would literally cut it right in the middle. So that would create blunt ends. Okay, so restriction enzymes can either create sticky ends or blunt ends. Okay, when they cut a specific piece of DNA, and they always have a specific recognition sequence in restriction site. Okay. So let's do a few review questions. Genetic engineering requires certain steps. Put the following steps in the correct order. We discussed this in the beginning of the lecture. Which one is number one? Yes, identification of the molecule. So it could be erythropoietin, for example. Okay. And next, this is first. Second is isolation. Yeah, so this is second. Isolate the piece of DNA that you are looking for. And third, manipulation. And fourth is the harvest and production, right? So that's the chronological order. Okay. All right. What naming designation is used with restriction enzyme? Give me the four things that are put into a name. Genus, yes. First is genus. Second, species. Third, strain. And last is the Roman numeral indicating the chronological sequence in which it was isolated. Okay. All right. Why are only type 2 enzymes used for cutting the DNA? What were some of the reasons? Type 1 and 3 have some issues, right? First, their restriction, their recognition sequences don't match the restriction sites. Kathy says don't need ADP, ATP, adenosine triphosphate. Okay, that's the source of energy, right? And also they have DNA modification activity, right? DNA modification activity. So that's not good. So that's why only type 2. Type 2 have all the right things, right characteristics. They cut where they hold. They cut where they hold the DNA. They don't need ATP. They only need magnesium, right, or a divalent cation, and they need they don't modify the DNA, right? Okay. Excuse me. What are the different ways in which a restriction site can be cut by endonucleases? Two different ways. Yeah, blunt and sticky ends, correct. Yes, that was Mercedes. Okay. Moving forward. Okay, now that we know how we can cut pieces of DNA, we need to know how we can analyze to see how we have cut the DNA. So let's say that you have this long piece of DNA, and oftentimes you would be cutting pieces of DNA that are circular. These are called plasmids, circular DNA pieces, small circular pieces of DNA. These are called plasmids. Okay, and we often find plasmids a lot in bacteria. Okay, so these are small pieces of DNA, and you can. So let's say that you have 3 recognition sequences in here. One over here, another here, and a third one right there. So now you take this piece of DNA and you digest it with these 3 different restriction enzymes. Okay. So hopefully these restriction enzymes are going to cut the DNA into how many pieces? So there's going to be one piece right here, another piece over here, and the third piece right here. So you expect to see 3 pieces, 3 fragments. So we call these fragments, right? So to be able to make sure that your restriction enzyme is actually working the way you expect it to, you can analyze the fragments by running it on a gel. Okay. Now, what is a gel? Gel is nothing but a piece of Jello. You know, see, at home you make Jello by pouring, by dissolving. What do you get? The Jello mix in water, hot water, and then you cool it down, right? The same thing with an agarose gel. So for analyzing DNA, we use agarose gel. So agarose is a polysaccharide that we obtain from seaweed. But it's a purified polysaccharide, right? So we get agarose powder and you dissolve it in boiling buffer. You dissolve it in boiling buffer. So and then you take the agarose solution and you can pour that solution into a little container, So it can form a slab. It literally form a slab of agarose, and when you cool it down, then it's going to form a gel, a slab of gel, right? And then, because it's a polysaccharide, what happens is that inside the gel is literally a matrix of polysaccharide molecules, long chain molecules. So you have a matrix, and you allow the DNA to migrate through this, what I would term an obstacle course. It's like an obstacle course, right? Now, if it is an obstacle course, lots of obstacles in the way, if I am smaller, I can run faster through the obstacles than if I am larger, right? So, because of that, you can literally separate smaller and larger pieces, right? The smaller pieces are going to run faster, and the larger pieces are going to run slower. Now, how do we make the DNA run through the gel? Well, it turns out that the DNA is negatively charged, right? DNA is negatively charged. So if I have a negatively charged molecule, I can have a negative electrode over here and a positive electrode over here, and I can have electrolyte solution here. So I run a current this way, right? I run a current. So what's going to happen to the negatively charged molecule? It's going to want to run to red, right? Negatively charged molecule is attracted to the positive electrode. So we say that DNA runs to red. If you remember that, you will not make a mistake when you operate a gel, right? Runs to red. Because when you actually set up the apparatus tomorrow, not tomorrow, but on Monday, you will be, putting in the positive electrode, negative electrode, and you will be setting the gel in there. And sometimes people set it up the wrong way, and they wonder why they never got any bands, right? So it's because you want the samples to be on the negative side. The sample to be on the negative side, and then it will run towards the positive, and that's how you differentiate the different pieces of DNA. Okay, so well, I'll show you some pictures soon that will make this clearer. Anyway, so you start with the powdered agarose, you dissolve it in boiling buffer solution. It's important to completely dissolve the solution. So you will, in the lab, you're going to be microwaving it. You're going to be putting agarose in a flask, in an Erlenmeyer flask with some buffer in it. Okay, you think you're going to be using TBE buffer, tris borate EDTA, TBE buffer. And you're going to put the agarose in it and microwave it. Now you want to microwave it until you don't see any haziness at all, complete solution, right? And so you want to make sure that your flask is large enough so this solution doesn't boil over in the microwave, okay? So you want to make sure you have a large flask and your buffer and agarose in there. So you're going to be boiling the agarose by putting it in agarose solution by putting it into the microwave, okay? And once it is completely dissolved and it is completely clear, then you can then pour the gel. Okay. Now, you can pour gels of different concentrations. Okay, different concentrations. So we'll talk about that in a minute. But depending on the concentration, agarose gels can be used to separate DNA molecules anywhere from 50 to 25,000 base pairs. Okay. Smaller than that, you need to use a different type of gel. We could use something called polyacrylamide gel electrophoresis. So this type of gel electrophoresis, we call it PAGE, polyacrylamide gel electrophoresis. So PAGE is used for small DNA molecules and also for proteins. PAGE is also used for separating proteins. Okay, but for now, we are going to be focusing on agarose gels. Okay, agarose gels for DNA molecules greater than 50, less than 25,000. Okay, and then, well, once you have run the gel. Okay, so what happens is you have a gel like this, and you have wells right here. So you would put in your sample into this well, and then you will have electrolyte solution around it. You have electrolyte solution, and you have two electrodes, right? Negative and positive. So the DNA has migrated, and let's say that your one fragment has reached over here, one fragment is over here, and another fragment is over here. Okay, you have three fragments. Now, can you see the DNA? Well, DNA is not visible to the naked eye. If you just look at the gel, you are not going to be able to see the DNA. So that's why you need stains. You need gel stains. Okay, and there are various kinds of stains that are used. Two common stains are ethidium bromide . And these stains, what they do is they intercalate with the DNA molecule and make them visible. And another stain is methylene blue. Okay. Now, ethidium bromide, you need UV light to make it fluoresce, make it visible. Methylene blue you can see without the UV light, but ethidium bromide is a much better stain. Methylene blue is less efficient. Okay, so most of the time we use ethidium bromide. Now, in the past we used to run the gel and then stain it, and ethidium bromide is kind of nasty chemical that we don't really want to use in the lab. So what we do nowadays is when you're preparing your agarose solution, we actually add a stain into, so when you're preparing the agarose solution, we add in a stain called CyberSafe. Okay. Into the agarose solution, so you don't need to stain the DNA later on. The stain is already in there, right? And so CyberSafe is similar to ethidium bromide, and it will show up under UV light. So after you have run the gel, you will be able to put it on a UV transilluminator. Okay, and you will be able to visualize it that way. Okay. So it's just a box that has UV light in it. Okay, so let's move forward. Are we clear on how we analyze the DNA we use? So here is a picture. Okay, so what I have here is the gel box. This is called the gel tray right here. This whole device. This is the gel tray. And what it has is it has two plastic sides, and the bottom, plastic bottom. And on these two sides, you have these gates. Here is a gate. So gate is like a plastic piece that can be screwed in. Right? There are two screws, one here and one here. So when the screw is tight, the gate is fixed, and this becomes like a little pan. This becomes a little pan, and you can pour the liquid in it, right? But when you unscrew the screws, then this gate can fall down, and so you can have liquid. You can have current moving through the gel. Is that making sense? So you will see it on Monday anyway. But essentially, you have a box. Initially, you have a box. You have made a pan that you can pour your agarose into. Okay, liquid agarose. And when you pour the agarose into the pan, you also put this little comb in there. This comb into the you set the comb in here, and see the comb has these teeth in it. That's what is going to create the wells, the sample wells in your agarose gel. Okay, so the comb gets set in there. You pour the agarose solution, you let it cool down, and then you can remove the comb, and that will create the wells inside the gel. Okay, and then once the gel is set, it is like Jello, right? Solid. Then you can drop the gates and place the gel tray in the gel box. Here is your gel box, right? So here is your gel tray that came from here. That's the gel tray. Okay. Now you cover the whole thing with electrolyte. Electrolyte, in this case, it's a buffer called TBE buffer, tris borate EDTA buffer, because it will carry the current. And so here is the negative electrode, cathode, which is black, and here is the anode, positive electrode, which is red, right? And so now you have placed the gel in the gel box, and you can introduce your samples. So you're going to introduce samples in these wells, the DNA samples. Okay. So this is what it looks like from the top. This is what it looks like from the top, and this is what it looks like from the side. Okay. So from the top, you can see, in this case, there are 6 sample wells that have been loaded with the samples. 1, 2, 3, 4, 5, 6, right? They are in a buffer, and you have the power supply, which is the negative electrode is connected to the negative side of the power supply, positive to the positive side. And once you turn the power supply on, usually we would run this around 110 volts, sometimes higher. And we run a current of about 35 milliamps. And so the DNA is going to start moving that way, and it's going to be off to the races. And then you will have the smallest molecule. If you look at it from the side, the smallest molecule is over here, and the largest molecule is over here, right? And so you will end up with bands. So you will end up with bands that look something like this, right? Depending on how many fragments you made by the restriction enzyme. If you made 2, you should see 2 bands. Now, what will happen if you just run uncut plasmid DNA? Uncut plasmid DNA. Where might you see that DNA? Correct. So Victoria says you should see just at the top, one band just at the top, right? So if you're running uncut DNA over here, you might see one big blob over here, uncut DNA, right? And if you have cut this DNA into 3 fragments, then you might see 3 bands, okay, depending on the sizes. In this in this lane, like that. Excuse me. Okay. So does that make sense? So this is what it actually looks like. We are going to use this in the lab. Can you tell me what this is right here? Anybody? That's the gel tray, right? Yeah, that's the gel tray, correct. And this is the comb. This is the comb, okay? And these are the screws right here to tighten the gates. The gates go along this side, okay? So, so the screws tighten the gates that make it leak tight. And you want to make sure that your gel tray is leak tight before you pour the agarose solution in it, okay? Otherwise, it will start to leak out, and you don't want that to happen. So before you pour the agarose, you want to leak test it with just water or buffer, okay? Okay, here is your gel box, and there is a platform inside it on which on top you place your gel tray on top of this platform, okay? After you have poured the gel. And then you don't forget to lower the gates, otherwise no electricity flows through the gel, right? Okay, and then here is the top of the gel box, which literally, see these electrodes right here? This plugs into that. Okay, and here is your power supply right here, power supply. Okay, you will see it next week on Monday. All right. So, how do the molecules behave in agarose gel electrophoresis? Well, they both DNA and RNA molecules run to red, and smaller is faster. And this is the approximate size that it is normally used for. Okay. Okay. Now let's talk about agarose concentration. Okay. Remember, we are going to make agarose solution, right? We are going to make agarose solution, and you can make different concentrations. So 0.8%, 1.5%, 3%. Okay. What does that do? By the way, if I say I want 1% agarose, what do I mean by that? If I say I want you to pour 1% agarose gel. Remember we talked about concentrations last semester. So the 1% is the 1 gram in 100 milliliters, right? 100 milliliters is weight volume percent. 1 gram in 100 milliliters, okay? So 0.8% would be 0.8 grams in 100 milliliters. So if I ask you to make 300 milliliters of 0.8% agarose gel, what you're going to do is you're going to figure out how much agarose is needed. It's going to be 300 milliliters times, there you go, Victoria figured it out, 0.8 grams per 100 milliliters, right? So that's going to be 2.4 grams of agarose, right? So you're going to weigh out 2.4 grams of agarose. You're going to get little less than the 300 milliliters of buffer, okay? Less than 300, so maybe um, like, 250, 240 milliliters of buffer. You're going to put the agarose into the solution, into the buffer in a flask, of course. And if you are going 300 milliliters, you might want a 1-liter flask. Okay, so you have a flask, maybe a 1-liter flask, because you don't want that solution to boil over, remember? So you put in your buffer, add the agarose, and then you're going to put it into the microwave. Maybe start with 1 minute and see if it is boiling. If it doesn't boil, give it another 30 seconds. Keep on doing it until you see the solution boil. And then take it out of the microwave. You're going to swirl it to make sure that everything is in solution. You don't see any hazy lines in here. Okay. Now, one thing to make sure of is that you use buffer and not distilled water. Okay. You use TBE buffer. Sometimes people forget and use distilled water to make the gel, and it doesn't work well. You get smudgy bands, not real nice bands. Okay, so you have to have buffer in there to conduct the electricity well, right? Now, what's the advantage of using lower or higher concentrations of agarose? When would you want to use lower concentrations? Remember, the higher the concentration, the tighter is this obstacle course. The tighter is the obstacle course. The lower the concentration, the gel is freer. Molecules will move easier through it. So when would you use lower concentrations, and when would you use higher concentrations? Well, when you want to separate relatively large pieces of DNA, then you might want to use the lower concentration, because it will be easier for the DNA to move through. And when you want to separate small pieces of DNA, really small fragments, then you want to use higher concentration, right? So the concentration of agarose affects the mesh size, and so higher concentrations are used with smaller pieces of DNA, and lower concentrations are used with larger pieces of DNA. Okay, and if you go more than 3%, the gel is just too concentrated, and it's not practical. And what would happen less than 0.8%? The gel is too fragile; it will break up easily. So a practical range is from 0.8 to 3% of agarose. Okay. Any questions about agarose concentration? And when you would use larger and smaller? Does that make sense? All right. So this is a picture of the whole assembly that I showed you before. I showed you the different pieces. Right now it's all assembled. Here is your power supply. Okay. The red line going to the positive electrode right here. The black line going to the negative electrode. Here is your gel box with the buffer in it, and your gel tray is right there, right? And hopefully the person who's running the gel has let the gates down so that electricity can flow through there, and the DNA is moving that way. Okay. And so this is what a gel might look like. Now, whenever you run a gel, you always run a standard with it. So, for example, a very commonly used standard is called lambda DNA. Lambda DNA. What is that about? So lambda refers to lambda phage. It's a particular type of phage that infects bacteria, right? So it's lambda phage. So it's the DNA from lambda phage that they use. So we know what lambda phage DNA, how big lambda phage DNA is, and if you cut the lambda phage DNA with Hindi3. Anybody know what Hindi3 is? It's a restriction enzyme, of course. So if you cut lambda phage DNA with Hindi3, we know that you're going to get these fragments: a 23,000 base pair fragment, a 9,000 base pair fragment, a 6,000 base pair fragment, a 4,000, 2,300, 2,000, and a 564. So we know that if we cut DNA, lambda phage DNA with Hindi3, you are going to get these known fragments. So this is our standard. This lane is running a standard. This is confirmation that our gel is running okay. So, so when you see these 7 bands, it means that you have run the gel correctly. Okay, and then it also helps you to figure out what's happening with your DNA. So here are samples of your DNA. This might be uncut DNA, and this is the DNA that has been cut. Right? So sample 2 has uncut DNA in it. Sample 3 has DNA that has been cut. Right? So now you can actually, what you can do is make a graph. And so on one axis you can plot the distance. You can measure the distance from this point. You can measure the distance of this fragment, this fragment, this fragment, this fragment. So you can plot distance. And over here you can plot the base pair number, right? The size. So you're essentially plotting size versus distance of the fragments. And so the larger size has shorter distance, so you're going to get a graph, something like that, right? Using this standard. So this is kind of a standard curve for your DNA, isn't it? And so let's say that you have run your sample and you measure the distance of this fragment, and it turns out to be over here. So you can then figure out the size of that fragment. That makes sense? So having a standard, the lambda Hindi3 standard, helps you to figure out the sizes of the fragments of your DNA that you're analyzing. Okay? That makes sense? Okay. So anytime you run a gel, you're going to be running a standard with it, and usually it is this lambda Hindi3 standard, which has 7 bands in it. Okay. Some vocabulary, see if we know all of this. Gel electrophoresis, we know that it's the process that uses electricity to separate charged molecules. In our case, it's the DNA fragments, but it could be RNA or it could be protein on a gel. Agarose is a polysaccharide from seaweed that is used for gel electrophoresis. Polyacrylamide is another polymer that is used for smaller DNA molecules and proteins. Ethidium bromide and methylene blue, those are dyes that are used to stain DNA. Ethidium bromide shows the DNA, makes the DNA visible under UV light. Methylene blue is in regular light, but it's less effective. We didn't talk about high throughput screening. Don't worry about that. Okay, some review questions. What size DNA fragments can you use agarose gels for? Yeah, 50 to 25,000, right? If agarose gel material is labeled 1%, what does the 1% refer to? Correct, 1 gram in 100 milliliters of the buffer, right? Okay, and then what causes molecules to be separated on an agarose gel? No, that's not what Victoria said, restriction enzyme. No, restriction enzyme is used to cut the DNA. What causes the molecules to be separated in the gel when it is running on the gel? Okay, so Jerry says electric current. Yeah, that's one thing, because electric current makes the negative molecule move towards the positive electrode. But then they are separated in the gel because of their size, right? Different sizes. Larger size over here and smaller towards the positive electrode, right? So it's the charge and the size that are key in separating the molecules. Okay. Name 2 common DNA stains that are used to visualize DNA on agarose gels. Yeah, there you go. Methylene blue and ethidium bromide, right? We use something like ethidium bromide. We use something called CyberSafe. By the way, sometimes when people are making the gels, they forget to add the CyberSafe. What's going to happen if that happens, if you forget to add the CyberSafe when you're making the gel? After running the gel, you won't see any bands at the end of the lab, when you're trying to visualize the DNA, you won't see anything. So that can be very frustrating. So always remember, when you are making the solution, once you have the hot agarose solution and you're cooling it down, add the CyberSafe to it. Okay. I think we are at the end of the lecture. What questions do you have for me? Okay, so Kathy is asking, how do you decide which restriction enzymes to use? Okay, so what people do is actually when they are doing genetic engineering, is they construct these plasmids. They construct these plasmids. Okay, and they construct them with they literally make restriction sites, different types of restriction sites. Okay, on them. And so, depending on what your DNA looks like, okay, they choose different types of restriction enzymes so they can literally construct a vector that they want, right? So they might use one type of restriction enzyme to place the gene for your protein. So let's say the gene for insulin, okay, and then they might use another restriction site to paste in the gene for the antibiotic resistance. We are going to talk about this a little bit later. Gene for antibiotic resistance in there. Okay, so they can literally design the type of plasmid they want and use different restriction enzymes for different purposes. Okay, because you know the restriction sequence, right? You know the restriction sequence for each enzyme. So knowing your DNA molecule, what is the DNA sequence is, you can tell if they use this particular enzyme, it's going to cut it here and here. If they use a different enzyme, it's going to cut it in different places, right? So knowing your DNA sequence will help you to figure out which restriction enzyme to use because you know the activity of each restriction enzyme. And so depending on what your DNA sequence is, you can choose a particular enzyme that's going to cut it in the right place. Does that make sense? Okay. Other questions? Okay, so remember that we have a lab this coming Monday, and the syllabus has the exercise number from the book that we are going to do. I don't remember the exercise number now, but it's not exercise 1. Okay, so make sure that you read the right exercise number before you come, understand what we are doing, and have a flow chart ready, right? So that the lab will be more efficient. And of course, if you have questions when you come in, you can ask me the questions. Okay, and also, anything in today's lecture or any other lecture you don't understand, feel free to talk with me during the lab. I'll be happy to explain things to you. Sometimes it's easier to explain in person than over the online. So, okay. All right. So if there are no other questions, we are done with today's lecture, and I will see you on Monday. Have a good weekend. CC moderator, we are done. Thanks a lot. You're welcome.